Hello everyone, myself Abhishek Kumar, Group 316. My topic is the goals of protein. So let's start. The main goal of protein physics are to investigate the role of protein in various biological processes. They also regulate genetic function and metabolic process. In addition, protein play a crucial role in cell movement, mechanical work and energy. It means what the proteins does our body to gain the protein or to mechanical work and energy conversion. Enzymes facilitate metabolic function, DNA replication and protein synthesis. Regulatory enzymes known as allosteric enzymes control metabolic pathways. Contractile protein enable cell movement. It means the contractile protein cell movement will not occur. Protein also plays a key role in oxidative phosphoglycerine and membrane transport. Special protein in animal function is oxygen storage, transport, hormone regulation, and immune defense. Structural proteins provide support and protection to tissues, organs. Proteins are essential for storing, transferring, and decoding chemical signals of living system. They also serve as receptors and signal transformer in the body's communication process. Protein molecules are highly complex structure with specific spatial formation. The goals of protein physics include studying the structure of protein molecules, means what the structures of protein, which type of structures of molecules and supermolecular system, as well as stabilizing the correlation between primary and three-dimensional protein structures. Three-dimensional protein structures, we read about it further. Researchers aim of understood the mechanism behind genetic coding, protein synthesis and gene regulation. By exploring the physical mechanism governing protein function, especially enzyme activity, ultimately solving those challenges will provide insights into cellular organismal phenomena from a molecular and physical perspective. Now we will continue the structure of protein. Structure of protein made up of chains of amino acids. It means the structure of the protein is made up of some amino acid. With the help of some amino acid, classified number of amino acids in chain, like peptides fewer than 50 amino acids, dipeptides 2 amino acids, tripeptides 3 amino acids, and polypeptide more than 10 amino acids. Proteins more than 50 amino acids, typically 100 to 10,000 amino acids linked together. Protein chains are synthesized based on a specific bodily DNA. Amino acids are composed of carbon, hydrogen, oxygen, and nitrogen. There is a structural difference. Some macronutrients chains, these type of chains of fatty glucose. Then the example of carbohydrate and glucose will convert into the glucose units, and lipids, fatty acids, triglyceride, these fatty acids, and amino acids. Denaturing, alteration of the protein shape and thus function through the use of heat, acid, bases, salts, mechanical. The denaturing, the alteration proteins shape, size, and function. What the function through the uses of heat, acids, bases, and salts, mechanical aggregation. There is a diagram to understand the normal protein. After the uses of heat, acid, salts, and mechanical aggregation, the structure will be in the simple form denatured protein. Importance of protein. Protein plays an important role in information of protoplasm. Nucleoproteins are complex proteins and act as the carrier of hereditary materials from one generation to another. Enzymes are biological catalysts and they are also proteins. Hemoglobin is a protein, it acts as oxygen carrier. Theoretical and experimental investigations studying the structure of protein molecules and supermolecule system is fascinating. It involves both theoretical and experimental, means the study of the protein, the structure of the protein with fascinating It involves in theoretical and experimental both the approaches. Theoretical use compositional method of predict protein structure based on principle of physics and chemistry study. On the other hand, utilize techniques like X-ray, crystallography and nuclear magnetic research, which is known as NMR, is determine the actual structure of proteins. Means it helps in X-rays, crystallography. There different types of techniques like to determine the actual structure of proteins. Function of protein. Protein has a critical physiological function. Protein is primarily used in the body to build, maintain and repair body tissue. In the event that protein intake is greater than that required by the body for this primary function, excessive protein is converted to energy for immediate use or stored in the body as fat. It means the protein intake in the greater body for the body use primary function is converted to energy, means instant energy, or for gym, like we are exercising gym, that's why for that reason immediate use, or otherwise it is stored in the body as a fat form. Protein energy will be used only after other energy source like carbohydrate and fat are exhausted or unavailable. Temperature. Heat causes protein to denature. Higher temperature will set proteins more quickly and cause to coagulate. For example, like an egg is cracked into a frying pan. The heat causes the protein standards in the egg to uncoiled, means denaturation. More heat causes the protein standards to reform in a new bond, and the egg sets and form of a solid. It means coagulation. Then let's come to enzymes. Enzymes will speed up the chemical reaction time. It means they upgrade or they increase the speed of reaction time. The denaturation process, example, soaking meat in marinade containing enzymes, will speed up the time taken for the meat to cook when heated.